Honorable President, invited speakers, distinguished guests, master and PhD students, ladies and gentlemen, welcome to 7th Special Symposium of Inter-Islamic Network on Oceanography. I am Bülent Çavaş, Secretary of Inter-Islamic Network on Oceanography and moderator of this event. INOC continues its series of symposiums contributed by distinguished researchers working in the field of marine sciences to develop high-level knowledge and skills. After this series of Inter-Islamic Network on Oceanography symposium and educational events, we see that the bond between researchers in different Islamic countries is getting stronger, and various collaboration projects proposals are formed in this way. I hope that this symposium will also provide international cooperation and collaboration among the researchers and scientists who work in different Islamic countries. Of course, INOC will always continue to act as a catalyst for the development of such collaborations. Today, we will have opportunity to listen to distinguished scientists who are conducting very important research in the field of algae. We are looking forward to listening to them. Before starting the symposium, I would like to kindly invite INOC President, Professor Dr. Sherman Açıkçınar, to make an official opening speech. Dr. Çınar, floor is yours.
Thank you. Dear scientists and participants, I would like to express my sincere gratitude and welcome you to the 7th latest developments on marine sciences symposium series that is one of the important activities of the Inter-Islamic Network on Oceanography. This event represents a significant milestone in our quest to explore the boundless potential of oceans, harnessing the power of algal biotechnology to address the challenges facing our world. I also welcome distinguished speakers from Turkey, Saudi Arabia, and Malaysia, and they come here to share their knowledge and experiences for the algal biotechnology. Algal biotechnology is one of the subgroups of the research under umbrella of biotechnology. In this area, micro or macroalgae-based bioproducts are widely investigated. The research on the algal biotechnology have been increased in recent years due to potential products which are used in many different industrial areas such as medicine, pharmacy, agriculture. Many of the Islamic countries have important aquatic ecosystems. Therefore, focusing on this area is of great importance from many perspectives. To contribute to this area, we wanted to organize this event to exchange the current studies from the countries. I believe that such networking activities will positively affect research opportunities and effective collaboration in the field of marine sciences among Islamic countries. I would like to thank the following institutions for their contribution to the realization of this event. COMSTECH, the Organization of Islamic Cooperation's Standing Committee on Scientific and Technological Cooperation, Dokuz Eylül Üniversitesi, Turkey, Celal Bayar University, Turkey, King Abdullah University, Saudi Arabia, University Malaysia Sarawak, Malaysia, University Putra Malaysia, Malaysia. INOC continues its activities by providing enriched trainings and supporting the research and development ecosystem in the field of marine sciences. In this context, we are pleased to see the development processes of projects based on scientific collaborations by different countries. You can check the INOC webpage and social media accounts to join the INOC network and get detailed information about research, education, and collaboration opportunities. I wish a very productive event with exciting and increasing discussions and exchange of knowledge so that together we can anticipate a future of groundbreaking knowledge, research, and technology for marine sciences. I encourage all participants to actively engage in the discussions, ask questions, and share your perspectives. It's an opportunity for us to learn from one another, to build networks, and to identify areas where our institutions can work together to enhance the quality of marine sciences. Once again, I extend my warmest welcome to all of you, and I look forward to enlightening and productive event that lay ahead. Thank you for being here with us today.
Dear President, Professor Dr. Sharmin Açıkçınar, thank you very much for your opening speech. And we can start our symposium now. But before starting, I would like to inform all participants that we will collect all your questions using this platform, I mean Zoom platform chat box. Please send them if you have any question during the presentation. Please send them to the Zoom chat box. We will ask them at the end of all presentations to keynote speakers. So I would like to invite first speakers. He is from King Abdullah University of Science and Technology, Saudi Arabia. His name is Dr. Claudio Fuentes Grunwald. Before starting his presentation, I would like to give you information about his academic background. Dr. Grunwald currently working as Algae Program Director for King Abdullah University of Science and Technology Beacon Development Program in Saudi Arabia. Claudio gets his degree in aquaculture technical engineering in 2001 by Arturo Prat University, Chile in 2008. He gets his Master of Science in Environmental Studies. Also, he gets in 2011 his PhD in Environmental Science and Technology. Both degrees from Universitat Autonomi de Barcelona, Spain. Claudio is an algal biotechnologist with over 27 years experience in design, construction, commissioning, and operation of microalgae plant facilities, with a long experience in microalgae growth in open and closed systems at industrial scale, such as photobioreactors, bubble columns, flat panels, raceways, among others. With an extensive knowledge and background in microalgae production, biochemical analysis, and metabolites purifications, including materials external to the cells across applications from aquaculture to medicine. He has more than 30 publications on microalgae, with half of his career working in industry developing on commercial novel technologies in microalgal biotechnology. Dr. Claudio, floor is yours.
Okay. Thank you very much. First of all, to Bülent and to Dr. Sharmin for the organizer of this event, and I really appreciate your time. So, can you see my screen?
Not yet.
Okay. There we are. Can you see it now?
Yes.
Okay, perfect. All right, yes, as I was telling, thank you very much to the organizer for this event and for the invitation to this event. And I would like to thank INOC, Dr. Bülent and Dr. Sharmin for this invitation. Because I am the first speaker, so the idea would be to give you an overview of algal biotechnology, starting from the very basic knowledge in algae. Right, just to let you know that here at King Abdullah University of Science and Technology, we are working with algae, seaweed, and microalgae since 2 years ago. This is an effort from the government, from the Ministry of Environment, Water, and Agriculture. And the idea of this project is to establish the algal biotechnology here in the kingdom, with the final aim to produce raw material for feed for animals. That is what we do. In my presentation, I will provide an overview in general of the algal biotechnology and what happened currently in the world with this technology. So starting from the beginning, so as probably everyone knows, algae is a really diverse group of photosynthetic organisms, can be micro or macroscopic organism, and they can inhabit a really different environment, from Antarctica to high mountains, desert, fresh water, brackish water, seawater. So they are everywhere. Basically, they are everywhere. The size, it can be just a tiny little few microns until several meters, as we can find in kelps in the northern Pacific or the southern Pacific Ocean, also in Asia as well. So far, there is more than 200,000 different species of seaweed and microalgae. Sorry for that noise, but it's some AC that is not working fine here. So basically, as I say, there is different seaweed and microalgae, and what it does, all these photosynthetic organisms, is basically they are absorbing CO2 by using nutrients and with the energy from the sun, and they create biomass and release oxygen. This is the basic about microalgae. Regarding this algae, what they do, why they are so important for our lives. So the first thing is that because they support all the food chain in the sea, right? So these are the primary producers. They support all small and big animals on the sea. And of course, probably everyone knows that they also have an important role in the climate by absorbing CO2 emissions and releasing oxygen. Actually, more than 50% of the oxygen that we breathe is coming from this organism. And millions and millions of years ago, especially cyanobacteria, they were the ones who created the atmosphere that we, and thanks for that, we are living now in this planet, right? So then, of course, there are some very good things as you have seen, but also there are some adverse effects on the environment, right? It will depending of which organism we are talking about. We can have several different effects, and actually later on you will see a presentation regarding a small group of algae that is called microalgae, that is called dinoflagellate. These dinoflagellate plus others, they produce several algae blooms. Some of them can be harmless, some other can be harmful, right? For instance, in this picture, you can see a harmful algae bloom that is damaging an aquaculture activities in southern Chile, right? It's a massive bloom. Also, we can see here, for instance, in the UK, is a massive bloom of Coccolithophoridots. And this one, what happened when they reach the maximum density and the bloom starts decaying, what happened is that deplete the oxygen and killing most of the animal around that area. But in terms of seaweed, we also have a massive effect, for instance, in tourism, in the Caribbean Sea, in Mexico, and all that area. There is massive sargassum bloom that every year show off to the coast, and then we have massive problem with tourism. So as you can see, we can have good thing about algae, but also, let's say, bad thing about algae, and we have to keep into that in account, all right? Then, how they are basically formed. So they have a different macromolecule composition depending on the strain, depending on the nutrient condition, and depending on the environment. But basically, we have the three main forms, which is lipid, carbohydrate, and protein. So in lipids, from the lipids point of view, Lipids are a part of the structural component, especially in the membrane, basically phospholipid in order to uptake whatever nutrients outside the medium to get into the cell, right? Then we have carbohydrate. Carbohydrates are basically energy, is energy, and they accumulate this energy inside the cell, but also they can exudate or they can release this polysaccharide as an exopolysaccharide in the medium, right? In seaweed, carbohydrate is the major fraction, and actually from carbohydrate we can get several different products, alginates, carrageenans, we can have agar, so different products from different fractions. And then finally, of course, we have protein, which is one of the most important. Protein, it has a structural component in different cells. And basically with protein, we can produce, in our case, for instance, here in the Kingdom or in the DAXA project, what we are producing is a protein for feed for animal by using a specific strain, spirulina, all right? Then the production system, all right, when we want to domesticate this type of algae, we have different production system, okay? As you can see on your left here, we have different type of system in the autotrophic mode, because algae can grow in microalgae especially can grow in three main different modes, all right? In autotrophic condition, by using the sun energy, they produce the biomass. We have photobioreactor, which are a closed system where you can control several parameters. You can actually produce pretty clean cultures. The issue or the drawback of that system is really high capital cost, all right? By contrast, you also can produce microalgae in open system, what we call, or raceway. As you can see in the picture below, those raceway are here in cows. We are producing spirulina adapted to Red Sea salinity in those one. And basically is way more cheaper in terms of capital cost, but also you can have an effect from the environment because it's open to the air. Then we have for heterotrophic cultivation, we use mainly reactor. It's a standard fermentation system. You can get more biomass, but also it's really critical in terms of the organic carbon that you are using, because you need to provide organic carbon in order to generate the biomass. And then a combination of autotrophic and heterotrophic is called mixotrophic cultivation, where you either inject CO2 and the nutrients, and also you inject organic carbon in there to create more biomass, right? From the seaweed side, we have different type of system, and also basically you can find land-based system, which are mainly tank, as you can see in the top image there on your right. You have this is our culture of Ulva lactuca, which is a standard green algae mainly used for bioremediation. You will see later on the statistic, but basically most of the production, seaweed production, is coming from wild catch. All right, as you can see, we have in the center a picture of a person in northern Chile, catching or harvesting kelps. In that case, is Lesonia. There are also sun culture, especially for Gracilaria, a rhodophyte, a red algae that is used for agar production, and is in that case they're farming in long base sandy beach, where they planting on the sun, and then they harvest. Depending on the season, especially in summer season. And the most the most common actually form of cultivation in seaweed base, especially in Northern Europe and in Asia, is long line cultivation. In this case, as you can see at the bottom right, this is a cultivation of Zacharina in Northern Europe, right? This is the standard long line system. So as you can see, there are different production systems, different way of produce microalgae and macroalgae. So then what we can get from this algae? So we can get several different products, all right? We can get animal feed, we can get cosmetic, nutraceutical, food, fertilizer, different type of fuels, fixers, and bioplastic mainly, all right? Seaweed are, as I say, are produced mainly by sea-based culture, and microalgae are mainly produced by land-based culture, right? So most of these products nowadays, they are in everywhere, basically. From seaweed, actually, this seaweed is everywhere, for instance, in cans, for instance, in toothpaste. They are gel and thickeners, so they use for food industry, for feed industry. We use microalgae for different human consumption, such as spirulina. So at the very end, there are several use of microalgae and seaweed biomass for different purposes. The beauty of this one is because they have a really nice fatty acid profile. They also have a really nice amino acid profile, and that is why they are so healthy for consumption, not only in human, but also in animals. Okay. So then what is the current status of algal product development and application? So as you can see, there are some of them, they are early development, some other, they are advanced development, okay? In early development, we can take, for instance, biofuels, antifungal, phycobilin protein, lutein, some of the pigment that they are producing, right? We also have advanced development, and especially, for instance, in fatty acid, we know that there are several companies, especially big companies such as DMS. They are currently producing an integrated DHA produced from Schizochitrium to salmon farming, right? So that is why they are using this omega-3 and omega-6 fatty acid in order to incorporate in salmon farming, right? We also have different products already in the market. You can buy by Amazon different tablets from chlorella, different spirulina products. So at the very end, there are several different products. All right. Then regarding the statistics, this is quite important, and this is something that we need to mention. As you can see in this FAO report, more than 32 million tons in 2020 was produced, or in 2019 was producing, and it's coming from seaweed, mainly from China and from Asia, right? The most important countries that are producing seaweed nowadays are Indonesia, Korea, China, Japan, because they have a long tradition and culture of eating this seaweed. That is why most of the production, over 32 million, is coming from those countries, but also we have production in Northern Europe, in South America as well. And most of the seaweed production actually is coming from wild catch. So that is important to say, and it's important to remark. All right, in terms of microalgae production, then this is a different story, also dominated by China, but in this case, the main strain that is producing worldwide is this cyanobacteria that is called Arthrospira, or we have, or Linospira now. Nowadays, we have different species, Arthrospira maxima, platensis, major, but mainly this algae is producing in open raceway, and especially in China and in some other countries. As you can see here, we have people that is actually harvesting seaweed, and in that case in Chad, also they are harvesting spirulina, right? Then where to produce, in this case, microalgae, because as I mentioned, basically seaweed is producing and is already harvesting from wild catch and from long line production in Asia, Northern Europe, and South America. So most of that one is already known, and they are producing a lot of this seaweed. But in terms of microalgae, is a little bit different, because it's all produced in land-based, in really, let's say, in a way, sophisticated system compared to seaweed. All right. And the question is where to produce microalgae for the future of algal biotechnology. There are two main reasons: climatic and biological reasons. Why? So as you can see, this is a paper that was published in 2022, and as you can see, the annual microalgae biomass production worldwide, depending of the solar irradiation, all right? And the annual protein production. As you can see, the main country that they can produce is those countries that we call the hot belt country. For 30 north latitude or 30 south latitude are the main countries. In this case, in the graph A, marked as red, you know, we can produce a lot of microalgae. Actually, just to let you know, this is a model, but in our facilities here at CAUS, we have been validating this model and actually it's fit pretty well. We are in the range of 60 to 80 tons per hectare per year, which is the right number in there. And also you can see that in Europe, for instance, the main producer or the larger scale producer in Europe are in southern Spain or southern Portugal, which is much specifically with this graph too. So this is pretty accurate, and we can confirm that the amount of algae that we are producing here in KSA in Saudi Arabia is matching the model. So that is why we have that climatic condition and the biological reason. Why? Because we have most of or a lot of sun, right? If you compare with northern country. Then, of course, land availability, because we have plenty of land here and it's quite cheap land, all right? Especially in the MENA region and the GCC region, MENA, Middle East, Northern Africa, and the Gulf Cooperation country. So all of them desert country. Also, land cost is nothing. So the amount of money that you pay for 1 square meter here is nothing compared to Europe or United States. All right. This is key point, energy cost. Energy cost is one of the key points. Current data regarding the price of a kilowatt in Europe nowadays is around 20 cents of euro, 1 kilowatt. The same kilowatt here in Saudi Arabia is less than 1 cent of euro. So in terms of operation, you can imagine that it's way more cheaper to produce in this country rather than in Europe or in the United States, for instance. Okay, and then the workforce cost, because the amount of money for payment for standard unskilled working in this country is lower compared to European country. And finally, legislation, right? Because it's way easy to grow in this country, despite that we don't have a proper regulation for algae. Anyway, you can produce algae here, and quite soon we will have a brand new regulation adopting from Europe, China, and the United States. So this is one of my last one. For the reason explained before, what we can see in terms of microalgae production for future algae technology, the MENA country or all desert countries in South America, northern Chile, southern Peru, in Australia, in all northern Africa, all in this area, we will be large-scale algae producers. Also, we will produce both algae commodities, all right? Of course, CO2 capture will be one of the key targets, especially in the GCC area. And what we call this is high volume, low value production, right? If we compare it with Europe, all right, for instance, in that case, in our point of view, there will be algae technology provided, but most of the technology is coming from Europe. All right, so that is why they cannot compete with us in terms of production, but also they can provide all the technology, and also they can provide the scientists, the staff, so they can train the people and then spread out in the world to develop this technology in all other countries. They are high volume metabolite producer, and actually what they call is low volume, high value, right? Low volume in terms of the amount of water that they produce, but they produce high value metabolites. So that is the picture that, in my opinion, it will guide the future of algae technology. All right, thank you very much for everything, and I hope I have done this in 15 minutes or less. I don't know. Thank you very much.
Dr. Claudio Grunwald, thank you very much for your very informative presentation. And I believe that this kind of information will provide extra insights to develop further research opportunities among the researchers and that are interested in this field. Thank you very much once again. I would like to invite the second speaker, who is Ergun Taşkın from Manisa Celal Bayar University. Ergun Hocam, I think he is sharing his screen. So before he is starting, I would like to give you information about academic background. Ergun Taşkın is a professor at Manisa Celal Bayar University in Turkey. He earned his PhD in hydrobiology at the same university. He has studied taxonomy, ecology, and inhibitory activities of marine macroflora, as well as marine monitoring and ecological quality status of marine coastal waters in Turkey and the Mediterranean Sea. He has developed ecological quality indices such as Marine Floristic Ecological Index and Benthic Ecological Assessment Rapid Index for the ecological quality classification of marine coastal waters by benthic macrophytes. He co-authored the books The Mediterranean Cystoceria, Ecology, Marine Algae of Turkey, Baufisse, and List of Turkish Algae. He has a scuba diving license. So, floor is yours, Dr. Taşkın.
Thank you, Professor Javash, for your presentation. My presentation on the marine algal diversity, their ecologies, and economical uses of them in the Eastern Mediterranean Sea. You know that seaweed or macroalgae are plant-like, but they classification under three main groups: brown algae, red algae, and green algae. Turkey has a different coastal geomorphological and physicochemical parameters on the coastal waters, such as salinity is different between Mediterranean or Aegean coast from the Black Sea or Marmara Sea. Another difference, marine macroflora distributed to 20, 25, or 40 meters depth in the Aegean or Mediterranean coast, while 10 or 15 meters on the Black Sea. So we listed about 571 macroalgal species from the Marmara Sea. It's followed by Aegean coast, Mediterranean coast, and Black Sea coast in Turkey. Brown algae have about 1,500 species in the world, and some brown algae are known as marine forest, such as Sargassum or Cystizoera. Have a look for these pictures for Facies Cystizoera, Padina, and or Siphonius Schistosiphon lomentera in the Mediterranean coast. And red algae has unicellular or multicellular species. This Sahlangia is sensitive species, Porphyra, or Calcareous phytomatiliton. Another calcareous species, Titonoderma, is common on the Aegean coast in Turkey. Another calcareous species is also Mesophyllum alternans or filamentous species Polysiphonia. These pictures from the Aegean coast in Turkey, it contains Mesophyllum alternans, and calcareous red algae is common on the Aegean coast. Green alga species unicellular or multicellular, and Some species is photophile and opportunist or tolerant, such as ulva or cladophora, or another species is syaphil or syaphil and sensitive taxa, such as codium and acetabularia, is common on the Asian coast and Mediterranean coast. Have a look for this codium bursa is common on the Asian coast, and acetabularia, acetabulum. Another green algae, Cephonius taxa, Caulerpa, and Bryopsis are also common on the Mediterranean coast. Marine algal taxonomy is started in 18th century by Max Baum. He listed 10 species from Turkey, and recently we listed about 700 taxa from Turkey. This numbers of brown algae, red algae, and green algae. And in the Mediterranean Sea is known about 1,170 taxa, and in the whole world about 10,000 species are known as macroalgal taxa. So this about 60% species is known in Turkey from the Mediterranean Sea. Italy has a highest species and it's followed by Spain and Turkey in the Mediterranean countries. We have also several invasive and alien species in the Eastern Mediterranean Sea. Have a look for invasive species such as Codium fragile is common on the Marmara Sea. Caulerpa cylindrica is common on the Asian coast or Mediterranean coast. Caulerpa scalpelliformis in the Antalya, in the Mediterranean coast, and Asparagopsis species is also common on the Mediterranean and Asian coast. Marine macroflora split into two main ecological groups. First group from Cystizoera, Posidonia, Padina, Cimodocea, some sensitive marine macroalgae, marine forest, and seagrass, while second groups opportunist and tolerance taxa, such as ulva, cladophora, or another filamentous marine macroalgae. We studied in all Turkish coast, and we found different ecological quality status on the coast, such as Marmara Sea is very impacted by industrial or urbanisms. Have a look for this quality, poor or moderate, generally in the Marmara Sea. Here is Black Sea, such as Zonguldak, Ereğli, and Samsun, Trabzon is also poor or bad or moderate quality status. Izmir is also bad or moderate quality status. Fethiye and Mersin Bay are also poor or moderate quality status. And economic uses, we have been studied on bioactive metabolites to find some marine macroalgal species. We tested several extracts to food pathogens, and we found Corallina officinalis, red alga Corallina officinalis, the highest inhibitory to Enterobacter aerogenes. His picture, these pictures to Corallina officinalis against to Enterobacter aerogenes, and we found about 34 millimeters inhibition zone. Also, we studied on acetylcholinesterase enzyme activities, and we found two species, especially Petallonia fascia is very highest to activities on acetylcholinesterase enzymes. This also Cystizoera crinita and Codium fragile is also very have antiprotozoal activities to Trypanosoma cruzi and to Plasmodium falciparum. We also have been studies to cytotoxic activities, and we found very important species in Turkey. Spiridia filamentosa showed strong cytotoxic activity against several sun lines. And we also studied two antioxidant activities, and we found also highest antioxidant capacity, Sargassum acinarum. Have a look to these figures, and very highest antioxidant activities in Turkey. So I tried to summarize biodiversity, ecological patterns, and economical uses, especially medicines and pharmacy from the Eastern Mediterranean Sea. Thank you for your attention. Best wishes.
Dear Dr. Taşkın, thank you very much for your very nice and interesting presentation. I believe that the information you provided in your presentation will allow us to think once more for the research opportunities in this field. So once again, thank you very much for providing us about your research information. I would like to invite the next presenter, who is Dr. Teng Sing Tung, University Malaysia, Sarawak, from Malaysia. Dr. Teng Sing Tung, yeah, I think he is sharing his screen. So before he is starting, I would like to inform you about his academic background. Just a second. Dr. Sing Tung is a senior lecturer. He began his academic career at University Malaysia Sarawak in Malaysia in 2016 with a major in aquatic science. He holds a Bachelor of Science with honor and Master of Science in Marine Biotechnology from University Malaysia Sarawak. He obtained his PhD Aquatic Science from University Malaysia Sarawak in 2016. His research interest is harmful algal blooms, which focuses on taxonomy and molecular systematic of harmful algae. Sing Tung was contributed toward the taxonomy and diversity of pseudo-niches in the tropical Western Pacific region, a region with scant information of ASP when he was still working on his PhD. His research activities are including taxonomy and molecular systematic marine and freshwater plankton species. So, Dr. Teng Sing Tung, floor is yours.
Hello, can you hear me?
Yes.
Yeah, good evening, everyone. Thank you, Professor Bulens, for inviting me to come to share my project. Okay, one of my project in my lab. Okay, so today I would like to share my project title with the marine small dinoflagellate and its impact on the aquaculture industry. So here actually is a negative impact, which is we focus on the harmful algal bloom. Okay. So what is a harmful algal bloom? Harmful algal bloom is the group of these algae that cause a negative impact to the marine organism. So today we will focus on the harmful small dinoflagellates. So what kind of what is a genus that actually fall into the harmful small dinoflagellates? So here we have the Heterocapsa, Carenia, Calodinium, and etc. So this is a three genus that actually is a very famous genus that cause a fish kill and also the shellfish kill. So come to the fish kill, actually the fish kill event is always related to the biotoxin and also this we call it as a hypoxia and anosic condition. So today we were going to focus on one small genus, which is the Calodinium. Okay, Calodinium, just a short introduction on the Calodinium. So Calodinium is an armored dinoflagellate. So it's a mesotrophic and marine small dinoflagellates. And why this Calodinium actually have the less attention compared to the genera of Carenia and the Takayama in the family Careniaceae is due to its smaller size. Okay, it's in between like 5 micron to the 15 micron. Okay, so first discovery actually is in the 1950, listed as a potential toxic algae and capable to produce the toxin like the calotoxin and the chamitoxin. So this is for the genus of Calodinium. So this is a key features that actually we use to differentiate the Calodinium. The first one is you can see here. There are three closely related genus. Okay, and for the Calodinium, they have the straight unique character which is a straight apical groove. Okay, this is a straight apical groove, and the presence of the ventricle compared to the this is the two different characters that actually you can find to differentiate the Calodinium from the Takayama and the Carenia. So okay, so come to the Calodinium. So, okay, Calodinium, we have a few species which is the toxic species. We have the Calodinium amiga, Calodinium australe, and Calodinium azanzi. Okay, so today we will just focus on the Calodinium australe because in Malaysia we actually face a big problem in the Calodinium bloom, especially in the Strait of Johor. Okay, Strait of Johor. So this is some chronology about this history for the Calodinium australe in the Malaysia. Okay, the first discovery of the Calodinium australe is in the Aimang Island, which is in the Malacca Strait. So at that time, actually, we don't have any bloom for the Calodinium australe, and so we don't have the fish kill report in that finding. And in the 2014, actually, we have the very huge bloom on this Calodinium australe, and it's happened in the Tanjung Kupang, which is located in the Strait of Johor, and that caused a mass kill of the cash culture fish. So at the right hand side, you can see the massive fish mortality in the cash culture fish. So in the 2016, actually, you can see the recurrence blooms. Actually, we discovered another bloom in the 2016. Also, the species is a Calodinium australe and caused a mass fish killed for the cash culture, and including the wild fish and the crustacean. Okay, so you can see actually some of the effects actually you can see on the on the fish. You see the skin slouching on the fish, and most of them are actually called barramundi and paddle snapper and the four-finger tree fins. So this is a fish that aquaculture fish in the Johor Strait, Malaysia. So this is a newspaper cutting about this fish kill that associated by with this Calodinium Australia bloom. Okay, so this is in the Johor Strait, reported in 2014. Okay, so you can see more than the 100 tons of the fish have been reported in the Johor Strait. This is including the Malaysia, Johor, and the Singapore, because we share the Johor Strait between the two countries. So this is one of the experiment that the fish bioassay that actually we conduct in 2016 in the field. I can show you some video here. So yeah, this is actually the simple fish bioassay that actually we conduct using the bloom sample, and we put in the small fish, and then you can see that the fish is swimming abnormally, and actually they can't really maintain the buoyancy, and yeah. So, and they also jump up and down, the behavior of this fish, okay, in the bloom sample, okay, the small dinoflagellate. And this actually happened within like 14 minutes to 20 minutes. We can see the effects of the fish already, okay. So, okay, next, distribution of the Calodinium Australia actually is very limited in the Asian countries. For example, it's only reported in Japan, Philippines, Malaysia, and Australia. So this is some of these SEM scanning electron microscope. Actually, you can see the morphological of this Calodinium Australia. Okay, so actually, the Calodinium Australia, the morphological character actually is fit very well in this previous study in the DCLS 2005. Okay. So these are the phylogenetic tree that actually we run for the last subunit and the ITS. Actually, there you will see that all the strand in the color of the Calodinium Australia from the Malaysia is actually clicked together with the original description of the Calodinium Australia with a strong bootstrap value. Okay, so actually we conduct the proper fish bioassay in the lab, and you will see that actually we have a few treatments. Okay, few treatments at the right hand side, the lice filtered cell medium and lice unfiltered cell medium, filtered live cell medium, and the live cell medium with medium, live cell with a medium. So meaning that for this live filtered cell actually is we lyse it, okay, we homogenize the cell, and then we filter the cell, okay, and we keep the medium for the experiment. And live unfiltered cell medium is we homogenize the cell and we without filter the cell, okay. And filter live cell, this is only left the medium. We throw away all the live cell, and the live cell with the medium means that it's normal cultures. Okay, control is only the medium without any cell, okay, without any cell. So you can see the mortality, fish mortality actually observed in the live culture medium compared, I mean early in the live culture medium compared to the filter and the live cell, okay. So meaning that actually when we homogenize cell, actually some of the toxin actually released to the cultures. And Calodinium Australia is more toxin after lyse, as it may be released the toxin throughout the water column. So why we want to actually conduct so many treatments here, because we want to make sure the fish actually killed by the toxin that produced by this collodium. And also we proved that actually the toxin actually released to the medium during the culturing techniques. Okay. So this is a fish bioassay that actually, okay, this is the work that actually done by one of my postgraduate students, and I can give some video here. And this is control, and this is the live filtered cell and the cell medium. So you can see the difference. The control, actually, the fish is swimming normally, and the live filtered cell medium and the live filtered cell medium is showing the same symptom as the fish bioassay that we conduct in the field in 2016. Okay, so the abnormal behavior is like that thing, and the fish will act like that thing and swimming erratically. And this behavior are similar with the fish behavior witnessed by fish farmer in the previous Calodinium Australia bloom event in the Johor Strait, Malaysia. So this is another treatments, which is a live cell with a medium and a filter live cell with a medium. They show the similar symptom. Okay, unbalanced swim, and you can notice that the petrol fins actually stop walking. You see, they are not even moving and they are sinking. They can't really maintain the buoyancy inside the water column. And this is a failure of the petrol fin in the swimming due to the calotoxin involvement. This is also reported in the DIPS ETL 2002. So following by this discussion, the fish bioassay, the toxin compound increase the rate of the gill damage because of hyperventilation in continuously stressed conditions. An acute response of the water-soluble toxin as a fish mortality occur in the less than 24 hours. And just for your information, our experiment is only conducted within 10 minutes because the fish start to have the symptom and mortality within 2 to 3 minutes or 5 minutes. Okay. So water-soluble toxin is typically associated with the acute response. Of course, this is reported previously. And this is a fish histopathology. Okay, this is a fish histopathology what that document in this study. Actually, we can see that damage to the gill, such as the hypoplasia. You can see the arrow here, and this is hypoplasia and lamella fissions, arrowhead. Okay. And you can see actually that this is a normal control, means that the fish gill, okay? And some of the other damage are like cell degradation. This is a 2 arrowhead, okay? Cell degeneration, and necrosis is a dot arrow, okay? And this is actually, this research actually show the involvement of the toxin from the observation of the gill alteration, and gill alteration in the fish treated with the calodinium, the calotoxin, KMTS, will hypertrophy, lamella fusion, necrosis, and loss of the secondary lamella. This is also reported in the DIP 2006, okay? And calodinium Australia filtrates cause more damage to the gill than the live cell and the culture medium because the filtered live cell is I mean that the lysis condition which we homogenize the cell will cause a more toxin that release to the medium. Okay, hyperplasia and the lamella fusion are the stage 1 damage, and the necrosis and the cell degeneration are the stage 2 damage. Okay, this is a level of the damage for the different treatments. Okay, in previous slide. So the conclusion here is the finding in this study conclude that the Calodinium Australia is a toxic dinoflagellate, and possible mechanism is Calodinium Australia is produce a potential ethiotoxin that disrupt the fish gill. So for the future recommendation will be more study need to be carried out to determine the detailed toxin structures and also the fish gill mechanism. because in this project we only look at these effects of the toxin toward the gill. Okay, we have no idea what is the impact to the fish, the other part of the fish. Okay, this is just some of the other research that we conduct in our laboratory. You can see that actually my group actually is quite actively in the taxonomies. Okay, the description of the new species for the Heterocapsa and also the Alexandrum, and you can see that actually we are very expert. That we are expert in the phylogenetic analysis, including the ITSU secondary structure, the CBC and hemi-CBC. And this is some of the work that actually we conduct for this Dolichospermum, which is the freshwater cyanobacteria. Okay, and you can find that the bloom actually is quite heavy bloom here. Okay, and this is the then ETL 2021. We published in the Plankton and Bendos Research, and about the green algae, which is related to the Botulococcus. As everyone knows, Botulococcus is the oil productions algae. Actually, the heavy bloom also can cause a fish kill in the freshwater. And this is some of the taxonomical work that actually we are in the progress. Okay, to describe the new species. And this is a references that I use in my presentation, and this is acknowledgement. And this is the campus. The University Malaysia Sarawak is a nice campus. If you have the chance to visit the Sarawak, Malaysia, then you can let me know. I can bring you to have a look in our campus. And thank you very much for your attention.
Dr. Singh Tung Tang, thank you very much for your presentation. It is really interesting and informative, and I strongly believe that the researchers from INOC member states will contact you to get more information and maybe to create a joint project proposal with you. Thank you very much once again for your presentation. So I would like to invite the next presenter, who is Emna Mehbi from Saudi Arabia. I would like to give you information about her academic background. Emna is currently working as a bioprocess lead for the Beacon Development Department at King Abdullah University of Science and Technology in Saudi Arabia. She earned her engineering degree in 2015, majoring in industrial biology and minoring in process engineering from the National Institute of Applied Science and Technology in Tunisia. In 2021, she completed her PhD in biological engineering. Recently, she integrated an important algae program at KAUST. She has been occupying a bioprocess lead for the KAUST algae program. Emna has gained experience in commissioning various industrial machines, including ultrafiltration, centrifuge, and high-pressure homogenizer. She led the commissioning process for these machines, ensuring their installation and functioning correctly to meet the requirement specification and produce high-quality products. Emna has extensive experience in scientific research with a strong background in modeling and process simulation, and particularly in the environmental field. She has achieved several notable research accomplishments, including the implementation of sequencing-backed reactor in semi-continuous mode for wastewater treatment. She has also established several new processes for bioelectricity production through a microbial full cell approach based on microalgae bacterial consortia. Emna has worked also as a process engineer in an EPC company, where she assessed the simulation of production unit of food concentrates livestock by Aspen Plus. Emna, floor is yours for your presentation.
Hello. Hi, thank you so much. So I will share my screen. Can you see it?
We see your screen, but not presentation.
Yes. All good?
Yes, you can start.
Okay. Thank you so much. Thank you for inviting us to present the principle of algae harvesting and downstream process. So first of all, I would start, today we will discuss the concept of harvesting. We will address questions related to what harvesting is, why it is necessary, and when, and how it is done. And we will discuss more about all the technologies used in the downstream process. First of all, let's begin by examining this picture, which visually depicts how cultivated algae can yield various products for the benefit of the world and the environment. Undoubtedly, there is much work to be done to produce diverse aspects and functions of algae biomass, like the production of animal feed, food production, biofuel, pharmaceutical product, cosmetic, and more. So here, to begin, we initiate, first of all, the cultivation. We initiate with the algae cultivation process by providing water, whether saline or fresh water, along with essential nutrients like CO2, exposure to light, in order to grow our algae. Subsequently, if required, we scale up the culture volume to a larger scale. Following this, the next phase involves the algae harvesting, which pertains to the separation or removal of algae from its growth medium in the upstream process. The conversion of this biomass into the desired end product is accomplished through the downstream processing, which comprises four essential steps. First of all, we harvest, we collect and separate the algae from its growth media, dewatering and washing, disruption or extraction, and then drying and refining, packaging, which is within each of these steps, numerous units operation can be used upon the specific requirement of the final product. We will address various questions related to the harvesting. This question includes the understanding of what is harvesting is, why it is necessary, and the specific timing of the harvesting conducted to generate diverse aspects of algae biomass products. So what is exactly harvesting? Essentially, it refers to the process of separation and collecting algae from its growth media. The methodology chosen for harvesting relies on various factors, such as the scale of the algae cultivation, the density and size of microalgal cells, and the specific requirement of the final product. So the harvest is about it comes after the cultivation process, and then we harvest. Then we go through a drying process or extraction process. Also algae harvesting can be the process of the harvesting and dry extraction can be mechanical, chemical, or biological. It's quite common also to combine those techniques to achieve higher separation rates at reduced cost. It's worth noting that harvesting, the harvesting of the algae accounts about 20 to 30% of the total production cost. So each of these harvesting methodology comes with its own unique advantages and disadvantages. The objective in downstream process is to prioritize the simplicity and energy efficiency to the fullest extent possible. Here we go into our second question. Why do we harvest? The foremost reason for harvesting algae is to cultivate the algae culture to potentially produce valuable goods. The algae cultivation, as we said before, is done to produce animal feed, products for human consumption, products for the environment. So harvesting stands as an important step that enables us to extract these valuable products from the cultivated algae biomass. Through the process of the harvesting, we unlock the full potential of algae cultivation, ensuring that the culture is put to a meaningful use and not wasted. Here we talk about all the volume that we have used during the cultivation. So when is the good time to harvest? The timing of the harvest is the crucial phase depending on various factors, including the type of algae being cultivated, the specific growth condition, and the intended use as well for the harvested algae. The first parameters and the most important one are the OT, the optical density, and the dry weight, that are significant indicators that shows us, for example, the optical density. A notable increase of optical density of our culture may signal that the culture has achieved the desired density for harvesting. Additionally, the dry weight measurement are often taken to confirm that the algae culture has reached the desired dry weight. This can be determined through periodic sampling and measurement. Also, here we can talk about the level of contamination, the stress, the pH, and the salinity. While monitoring the salinity, for example, particularly on a larger scale, becomes crucial. So harvesting is the decision of harvesting is made after the examination of different parameters, mainly dry weight, optical density. Let's talk about the stress and the nutrient depletion that must be managed, and harvesting should take place before this condition occurs. This is especially critical for extracting compounds like carbohydrate, protein, and pigment. Continuous monitoring of these different parameters are essential, and the timing of harvest should be adjusted accordingly. I think that's it about all the timing of the different parameters for the timing. So let's go here. Now let's explore how can we harvest algae in a good way. The methodology used to harvest algae can vary significantly based on factors like the specific type of the algae. It can be spirulina, it can be Nanochloropsis, it can be different Cimmeroli, different strains. And the scale up of the cultivation from raised phase, from photobioreactor, from small scale in the lab or outdoors cultivation. Here we need to know from our start of the harvest. Various techniques are used during the harvesting and downstream process. To begin, since algae cultivation necessitates substantial water use, so separation methodologies such as dewatering and washing come into play. This method involves the use of specific technology like flocculation, filtration, sedimentation, and centrifuge. Then subsequently, the downstream process refinement are mandatory steps if we intend to extract valuable compounds. Otherwise, we go to the drying process, which is where we can use freeze dryer, dehydrators. This is related to small scale, lab scale, and we can use the solar drying, sun drying, spray drying when we are working in a larger scale. So we should note that during the harvesting or during the downstream processing, the concentration of algae biomass depends on the increasing of factor concentration. It means that when we start from the dewatering or washing, we need to go into a higher concentration factor, starting from our culture that it's between 0.2 to 5 grams per liter, up to 100 to 200 grams per liter. Here we go again furthermore into details regarding the harvesting in lab scale, where we can use different and affordable condition or techniques that we can use in small scale, starting from flocculation, sedimentation, coagulation, and sometimes centrifuge. And then we can go to second step for extraction or drying. And then, especially in lab scale, we go to analytical analysis. Furthermore, into a larger scale, which is more important because we are about to harvest massive biomass of algae. The methodology that we are using is mainly first of all dewatering and washing using filtration or centrifuge, and then we pass through extraction if you are about to extract or some valuable content from the algae biomass, or drying process, then storage. Just also to highlight that in the larger scale, we mainly use for the watering and washing ultrafiltration and centrifuge. And when it's about the drying process, we use solar dry and spray dryer. I just need also to focus on the harvest and downstream process should be on cost-effective and affordable methods in order to not spend a lot of money on energy and equipment. Last thing, so here we have reached to our conclusion in the downstream process. We should note that this many steps the downstream requires, but we should mainly start from the final product. So from our final product, we can set up the different techniques and methodology that we can go through the downstream process. And this is all depends on the aspect of our final products, whether powder, liquid concentrate, algae paste, frozen biomass, or even extracted compound. Also, just I should highlight that during my work here in downstream process, we should also not only work on the techniques and the methodology, but we always need to optimize how to reduce the lost biomass during the watering and washing and centrifuging as well. We need also to optimize our flow rate, inlet and outlet, the pressure for maximum efficiency. And also we need to implement effective storage strategy to preserve our harvested materials. Thank you, everyone. Thank you.
Dear Dr. Emna, thank you very much for your presentation. It is really interesting.
Thank you so much.
King Abdullah University of Science and Technology is a place, I think, for international researchers to conduct collaborative working projects. And I believe that you are one of the lead director and lead researchers to communicate with algae for further researchers. So thank you once again.
Thank you so much. Thank you so much for
the presentation. I would like to invite the next presenter. He is Levent Çavaş. I would like to ask him to share screen when I read his academic background. Levent Çavaş is currently the member of the Faculty of Science, Department of Chemistry, Biochemistry Division in Dokuz Eylül University. He obtained his PhD from Graduate School of Natural and Applied Sciences in Dokuz Eylül University in 2005, and he continued his studies and researches in Dokuz Eylül University. His main scientific interests are the biochemistry and biotechnology of invasive marine seaweeds in Mediterranean Sea, isolation and characterization of secondary metabolites, eco-friendly antifouling biocides, and paints and bioinformatics. He has 90 scientific papers published in SCI expanded journals with higher than 2,500 citations. He played roles in many European Union-based projects. He is one of the members of Science Commission in the World Underwater Federation. Floor is yours, Levent. Levent, we don't hear your voice.
I hope now you are hearing me, right?
Yes. Can you make your presentation full screen?
Okay. Now, do you see the second slide?
Yes.
Okay. First of all, thank you very much, Bülent. Bülent is my twin brother. Actually, I also thank Shermin because I witnessed your efforts to organize this kind of seminars. I believe that they are highly important, not only for Turkey, but also other countries under the umbrella of INOC. And also, I believe that this kind of seminars and symposium will contribute, provide a big contribution to the development of algal biotechnology in the member countries. Okay, in my presentation, I want to share my papers related to from 11 selected papers based on algal biotechnology. So I will start with the first investigation related to Caulerpa taxifolia. Okay, let's start. In our first investigation, we investigated the Caulerpa taxifolia. Actually, this is a kind of taxonomic studies based on the genetic parameters. So why I started with this publication? Because, you know, Turkey is surrounded by the seawaters, and also beside we have the algae species, and also our coastlines are under stress of invasive species of the Caulerpa genus. So this investigation was published in Eusterine Coastal and Shelf Science in 2007, and then we realized that we have also another invasive species in our coastline. And in our university, we wanted to create a subgroup within the chemistry department in Dokuz University to investigate the biochemistry and biotechnology of this invasive species. So therefore, I wanted to start with this investigation. Okay. After that, my colleague Ergün can understand me that the current name, systematic name of this species is Caulerpa cylindrase, but at that time it was one of the variety under the Caulerpa racemosa. So it is one of the invasive species under the genus of Caulerpa, and it is alien for Turkish coastline. So when we look at the papers before publishing before 2008, it is suggested that this species can be collected and burned. But we collected this species from our coastline, and we realized that there are very good functional groups, and then we wanted to convert this biomass into an adsorbent. So according to our results, which was published in Bioresource Technology, which is one of the top journal in the field of biotechnology, and our results showed that the adsorption capacity of this species against the methylene blue is comparable with the commercial products. And then, since this species is very important, we wanted to study the secondary metabolites, caulerpenine, because it is important for their adaptation to ecosystem. You know, it is an invasive species. And then we wanted to use queuing theory. It was the first contribution to the scientific literature, and we found out that the caulerpenine levels released from, secreted from the Caulerpa species can show fluctuation, and we found out this trend by using the queuing theory, by the contribution of the researcher from Windsor University in Canada. So after that, we wanted to use the Caulerpa racemosa, actually with the current systematic name Caulerpa cylindrase. We wanted to convert this biomass into an adsorbent again for the malachite green. Why we use the malachite green? Because malachite green is one of the important dye for textile industry, and unfortunately, due to the low, this malachite green is a chemical, and it is very resistant for the degradation in seawater. Therefore, it is needed to remove this kind of materials from seawater or aquatic ecosystem by using a very cheap adsorbent. So we thought that Caulerpa cylindrase could be a low-cost adsorbent material, and we carried out this investigation, and we found out very high adsorption capacity. For example, according to Friendly equation, we found out that the adsorption capacity is like 13. As you can see from this table, the adsorption capacity is based on the temperature that you study. So this paper was published in Journal of Hazardous Materials. And then we wanted to publish another study by using the Posidonia oceanica. My colleagues will understand that Posidonia oceanica is not an algae species, but I wanted to add this paper to this presentation because Posidonia oceanica is under stress of invasive Colerpa species within the Mediterranean Sea. Therefore, they are in some region of our beaches that we studied, we saw increased dead leaves on the beach. Unfortunately, these accumulated dead leaves of the Posidonia oceanica are collected by the local municipalities, and in order to get rid of these species, they are generally burned. Instead of burning this species, we wanted to use this species again, highly cheaper material, and we studied again its absorption capacity against methylene violet, and this paper was published in marine biotechnology with the high absorption capacities. After that, we wanted to study the secondary metabolites isolated from the Colerpa species. As I mentioned in the beginning of my presentation, Colerpa species, especially the alien one, are very important, and their success is based on the secretion of secondary metabolites. Actually, there are two dominant secondary metabolites. One of them is cholerpenine, which is based on sesquiterpenoid structure, as you can see here. And we isolated this sesquiterpenoid structure by using HPLC techniques, and then we wanted to test its inhibitory activity against lipoxygenase enzyme in human metabolism. And fortunately, we found very high inhibition activity by using cholerpenine, and we published our data in again marine biotechnology. So after publication of our cholerpenine-based study in marine biotechnology, and then some researchers from Friedrich Schiller University in Jena, Germany, contacted with us to carry out a joint project. And we also isolated cholerpenine, and we, let's say, modified the structure by using semi-synthetic methodologies, and then we obtained different compounds, different modification, different cholerpenine versions. Finally, we found that higher inhibition capacity with the newly synthesized cholerpenine derivatives, and this publication was the results was published in ChemMedChem. And then after these publications, we created a consortium and applied an Horizon project. Then we obtained an ITN project from the European Union. Actually, this paper is one of the result or outcome of the Horizon 2020 project. Catherine Lee Morrissey was I am one of the co-advisor of the Catherine Lee Morrissey from the South Africa, and we carried out this investigation. We found out very interesting results related to host specificity, thallus differentiation, which is very important and very important research question before 2020. We found very interesting results related to thallus differentiation and also bacterial communities on green seaweeds. One of them is Colerpa species, and we characterized the bacterial colonies and also communities on Colerpa prolifera and Colerpa cylindriaceae. So this publication was published in Frontiers in Microbiology. And then we thought that the biomass of Colerpa species can be used to produce functional foods, because we found that there are very important bioactive peptides within the tissues of Colerpa, and by retrieving the Rubisco enzyme sequences from the unit broth, and then we found that Colerpa species can be used to produce functional foods. Why? Because we found that there are angiotensin converting enzyme inhibitors. You know, this enzyme is responsible with the hypertension. So in case of you use GOLERPA-based tissues in your food, angiotensin converting enzyme can be inhibited. And also another important enzyme is the peptidyl peptidase 4. This is very important enzyme for, let's say, the diabetes. Therefore, when you use GOLERPA-based food, the peptidyl peptidase can be inhibited. Okay, in our another study, we thought that GOLERPA lentillifera can be used to synthesize the green silver nanoparticles. You know, silver nanoparticles have widely distributed utilization areas from medicine to electronics. Therefore, we wanted to synthesize by silver nanoparticles by using GOLERPA lentillifera. We synthesized very good silver nanoparticles. They are very stable and they can be stable like for more than 10 days. And also in our investigation, we wanted to test their let's say, the toxicities against the Allium ascolanicum. So we found that the toxicities is based on the concentration of silver nanoparticles that you use. So we have enough. We have silver nanoparticles in our laboratories. If anybody is interested in these kind of silver nanoparticles, we can share our materials. And this is our final contribution, which was published in Bioresource Technology. We studied Ulva lactuca. Ulva lactuca is a big problem for Izmir Bay due to the pollution. The bloom of Ulva lactuca is observed regularly in Izmir Bay, and unfortunately, the biomass, overgrowth biomass in Izmir Bay caused bad odor and also bad view in the recreational areas. Therefore, the people around the Izmir Bay is unhappy for this material. In our investigation, we wanted to convert this material in to synthesize silver nanoparticles, as we studied in our previous paper. But in this time, after synthesizing of silver nanoparticles, by using these silver nanoparticles as catalyst, we produce biohydrogen. You know, biohydrogen is very important material for the energy, and the biohydrogen is can be utilized in a car, especially as an alternative energy source for the electric-based cars. Because electric-based cars uses lithium technology. Lithium is a trace element in Earth. Therefore, the hydrogen-based cars can be developed in near future, we believe, and we aim to produce biohydrogen for these purposes, for the automotive industry. So these are the references for my publications. So thank you very much for your attention. If you have any question, I would be more than happy to answer them.
Levent, thank you very much for your very nice presentation and articles that you published in this field. I believe that you are ready to share these articles with the researchers if they want to get more information from you. So thanks once again for your presentation. I would like to ask the next presenter, who is Gabriel Ivan Romero Villegas from CAUS. Before his presentation, I would like to give you information about his academic background. He holds a PhD in Industrial Biotechnology and Bioprocesses applied to agri-food and environment in 2018, Universitat de Almeria, Spain. He has been working with microalgae culture since 2011 in lab and big scale. He has 2 postdocs in Instituto Teknolojik de Sonora, Mexico, where he worked with native microalgae and their impact in germination process of seeds of agricultural interest. He has been working at King Abdulaziz University of Science and Technology as an algae lead in the governmental project development of algal biotechnology since 2021. in which is responsible of industrial scale-up microalgae cultivation. Dr. Villegas, floor is yours for your presentation.
Well, thank you very much for the presentation. Well, my name is Gabriel Romero. I will talk about how to grow microalgae, sorry. First of all, I need to say that Dr. Claudio gave us an interesting view about the status of microalgae in the world. However, I will talk about what we need in order to cultivate microalgae. This is going to be a brief resume about it because this is a big subject that must be explained with more details and more time. So first of all, what are microalgae? Microalgae are microorganisms that have the ability to carry out photosynthesis. They constitute an important source of biomolecules with a wide range of applications that can be exploited for commercial use. This as the same as plant, microalgae can produce different natural products. They have been used to obtain natural products of low value like biofuel, fish food, animal feed, or biostimulant. However, the microalgae market also includes high-value products which are used in the market of cosmetic, human nutrition, and pharmaceutical. In order to produce microalgae, we need to take into account some inputs and parameters that we need for the cultivation. Here, I'm showing some of them that those are light, water, nutrients, pH, and temperature. I'm going to talk just a little bit about all of them. Regarding to irradiance, microalgae need light as source of energy to do the photosynthesis. And the intensity of this one can have a big impact on how healthy your culture is going to grow. For providing light, we have 2 ways to do it. One is artificially, and the other is naturally. The first option is commonly used in indoor condition. When we cultivate in indoor condition, we usually use LED and fluorescent lamps. This is because we are cultivated. This is because when we are cultivating in outdoor condition, we want to manage the intensity of the irradiance received for the culture. If the culture receives more light than the necessary, we can cause photo high inhibition, and we can kill the culture. Or the opposite, if the culture doesn't receive enough irradiance, it won't grow properly. So the irradiance usually in indoor condition goes from 50 to 800 microeinstein. On the other hand, we have outdoor condition. This is important to mention that in outdoor condition, the irradiance will depend on the environment condition in that moment. That means, for example, if we have a clear day, we can have peaks of irradiance, or the opposite, if we have cloudy days, we can get a lack of irradiance, and again, we are not going to have a good growth. The next one is water. I need to mention that microalgae needs water to grow, sorry. And here we can talk about microalgae natural adaptation. Water is classified as fresh water, brackish water, and seawater. And we can find microalgae in all these environments. So microalgae are adapted to each of them. This is so important to know at the moment when we are thinking about what microalgae we need and we will cultivate. Because we need to think in the source of that water. For example, if we have fresh water, we need to think that we need to have near to us a fresh water source. This could be, for example, a lake, a river, I don't know, tap water, water wells, and the same for the other 2, brackish water and seawater. Regarding to nutrients, we can use a wide source of nutrients, but it will depend on the use of the biomass obtained at the end. For example, in laboratory condition, we usually use analytic grade salts. This is because we want the culture as clean as possible. We need to avoid the contamination, and we need to provide the optimal condition to the microalgae to grow. But on the other hand, if we use, if we want to scale up our culture, let's say an industrial scale, it is necessary the use of cheaper nutrient source. So we can switch to agriculture grade nutrients like fertilizers. On the other hand, we can use alternative source like wastewater. As we can see here, wastewater is rich in compounds like ammonium, nitrate, phosphorus, and carbonates that they are the main nutrients required for a microalgae culture. The next one is pH. As I mentioned before, microalgae are adapted to different environments. This is the case for pH. Microalgae can be found in different range of pH environment. For example, we have Cymrulae, which lives in acidic water with pH below 2. Another example is most of the microalgae strains lives in lakes, rivers, or any other lagoon where the pH is around 7 and 8. And finally, we have microalgae like Atrospira, which can grow in alkaline pH up to, it can reach 11, even 12. Another adaptation is the temperature. As many other microorganisms, microalgae are adapted to different temperature conditions. We can find psychrophiles, mesophiles, and thermophiles. We have the example here. We have saprophiles, we have Acodesmus and Chlorococcum that they are adapted to temperature below 20 degrees. We have Nanochlorosis gaditana and Chlorella, which they are adapted to temperatures around 21 and 39. And the thermophiles like Picocolorum and Cymrulae, which are adapted to hot temperature about 40 degrees. So we must have the ability, when we are cultivating microalgae, we need to have the ability to heat or to cool down or culture depending on the microalgae we are using in that moment. Regarding to carbon, carbon is one of the most important inputs to consider when we are cultivating microalgae. We can use several sources like CO2, sugar, like fructose, sucrose, maltose, and glucose. Even we can use flue gases. For example, CO2. No, however, there are some advantage or disadvantage on the use of this. For example, CO2 provides a high carbon fixation. However, it is too expensive for the process. The use of sugar can enhance the growth, but it exists a high contamination risk. In other microorganisms can take advantage of that carbon source in the culture, and they can they can become the main microorganisms in the culture. Regarding to flue gases, they reduce energy use, yes, but they may contain impurities like heavy metal that which may affect the quality of the algal biomass. Another really important parameter that we need to have in mind is the mixing of the culture. Mixing is really necessary to prevent algae sedimentation and ensure that all the cells in the culture are equally exposed to light and nutrients, and improve the release of excess of oxygen from the medium. There are several shades for bioreactors which try to improve the mixing of the culture. Here I show some of them. They consist in a column when the air is injecting on the bottom and is releasing at the top. These reactors have shown the best mixing in photobioreactors or in all the photobioreactors. On the other hand, we have the reactors that present two types of flows. They present laminar flow and tubular flow. On the laminar flow, we don't have a good mixing. So that means that we need to use something to help the culture to have a well mixing. So they have been adapted with an aeration system, this one and this one, that is basically a column where they inject air, and the air helps to mix the culture and to release oxygen at the same time. So it is important to have in mind a reactor with a good mixing system in order to improve our biomass production. Well, after mentioning all the basic parameters to have in mind for microalgae cultivation, this slide show the microalgae production process where we can find all the parameters combined. I think most of us have seen something like this. We have here our microalgae. We have our inputs, which are CO2 as carbon source. We have light, water with its nutrients, and we have parameters that we need to control, like pH and temperature. Okay, when we have cultivated our microalgae, this is processed through the downstream processes to obtain the biomass from microalgae. After processing this, we can obtain protein, lipids, and carbohydrates, which can be converted in products for agriculture, aquaculture, cosmetic, biofuel, animal feed, pharmaceutic, and animal nutrition. Now, I will explain a little bit about the scaling up process in a simple way. This is the schematic of scaling up process. Everything begins with the isolation of the microalgae or interest. This is done in a plate with agar and nutrients required for its growth. Once we have colonies, we take one of them and we put it in a container. In this case, it's a Erlenmeyer flask, where we add our culture media and the strain isolated. Once we have enough biomass, in this case, they say that we need to wait 1 week in every step, but it's not mandatory because in the real application, it must be longer than that. So in every step, what we do is we increase the volume of the culture. For example, here we have 100 milliliter. After 1 week, we transfer that culture to a bigger container. In this case, is 1 liter reactor. And this process will be repeated again and again until we have the desired total volume, that in this case is 100,000 liters. Well, here, once we talk about what microalgae needs, imagine that we want to get a product from microalgae. I had thought that we want to get, for example, lipids. Now we need to look for a microalgae who produce those lipids. I have read several papers. After reading several papers, we have that chlorella is one of them. Why? Because these have that up to 30% of lipids in the dry wet base. I know that chlorella grows in fresh water. Most of the researchers choose medium to grow it. We set the laboratory condition, in this case for light, 350 microinstants with artificial photoperiod of 12-12. It means 12 with light and 12 hours in darkness. We set a pH at 7.5 and a temperature of 25 degrees. So when we have the desired concentration in this culture, the culture needs to be moved to outdoor condition, where some parameters are not provided, when some parameters are provided by the environment. So we don't have control of them, like temperature. As you can see here, we in the laboratory, we set it at 25, but when we put it in outdoor condition, we cannot control this. So we need to adapt this strain or this culture to the host condition for it in the outdoor. Here we can see the process of, this is imaginary, this is not the culture that we are talking about, but this is the process of scaling up, that it goes for the laboratory scale to outdoor scale, when we have the column of 60 liter, then we transfer it to raise weight 800 liter, and then to a final volume of 1,000 liter. And here I want to show you the process that our team in DAXA have done about what we are talking about. This is the process of, this is the process of scaling up, where we started with a few milliliters in the laboratory, and then we scale it up, up to this volume, passing through different another volumes. For example, we have 200 milliliters, then we scale it up to 5 liters, then we scale it up to 60 liter, then to 1,000 liter, and finally we have here our facilities that we can manage up to 80,000 liters. And well, thank you very much.
Dear Dr. Gabriel, thank you very much for your very nice presentation and information. The information you provided in here, actually, it was a nice opportunity for us to listening them. And thank you very much once again for this opportunity. I see that the interaction is going on in Zoom chat box regarding the questions. As I mentioned in my in the beginning of this symposium, you can send all your questions to the Zoom chat box. I believe that the keynote speakers will answer them in there in order to keep our time. So as I see on the Zoom chat box, the interactions are going nicely. So thank you very much for your questions, and thank you very much for the answered by the keynote speakers. I would like to invite the next speakers, who is Dr. Tan To-hee. Before his presentation, I would like to give you information about his background. Dr. Tan holds a PhD in marine biotechnology in 2017 from University of Malaysia, Sarawak. Food safety is his first priority because readily available food should be safe for consumption. His research background was on phytoplankton associated with harmful algal blooms. Now he is a senior lecturer in University Putra Malaysia Bintulu Sarawak campus since 2019. He taught both diploma in fisheries and Bachelor of Science with honors in aquaculture. He looked forward to a world without starvation and everyone getting sufficient nutritious food, and hence started his research group, Sarawak Live Feed Research, with a career direction to focus on aquaculture organisms, live feed selection framework to improve fry survival, development, and quality. His current research interest is to find and utilize the potential micro and macro algae as live feed. Dr. Tan, the floor is yours for your presentation.
Thank you, Prof. Bulan, for the introduction. Hello, everyone. Good afternoon from Malaysia. I'm very excited to be here. So let me share about my sharing on this phytoplankton for live feed in Malaysia aquaculture industry. This is my first grant in this institution, and this is the beginning of my career as an academician. So I will start with the introduction. That the world, I think everyone agrees that the world is getting a bit crowded nowadays. There's a lot of people, and the number of human population keeps going up. And when there's a lot of mouth, that means we need a lot of food. So aquaculture is actually one of the industry that helps to meet the demand for providing sustainable seafood. And also in Malaysia, aquaculture is actually an important industry here to produce food for us to eat. So in the hatchery, food fish are produced in the hatchery stage, and also ornamental fish. So this is quite a big industry. However, in the industry, small fish larvae, actually their survival rate is quite low, and the quality from the hatchery, the local hatcheries, is very low. So live feed is actually one of the main factors that contribute to the successful of this live culture, of these fish cultures. So we are now looking at a phytoplankton, which is the vegetables, the plants in the natural environment. That is the food, the primary producers for this fish larvae. So in Malaysia, actually, the Fisheries Research Institute, they have started this live feed research since the very early 80s. They are focusing on unicellular green algae, diatoms. So what is the basic problem here is to supply sufficient live food of the best quality at the lowest cost upon demand. So why phytoplankton? Now, when you ask me why I do phytoplankton, I'll tell you that I'm being trained for phytoplankton. In my research, in my postgraduate studies, I study harmful algal blooms with Dr. Tang here, another speaker. We are from the same lab. So from that time, I was looking at all these harmful species. But when I come here to this institution and started my career here, I'm teaching fisheries and I'm teaching aquaculture. So I'm trying to fit into the niche. So I'm looking at now phytoplankton that are suitable for food, for fish. So why phytoplankton? Because they are the primary producers. They are the base of the food chain. They are the food for higher trophic level. And they have these proteins, lipids, vitamins, minerals. All these are very essential for the growth of this fish larvae for our food. And also, actually, phytoplankton, they are quite easy to culture. I would like to thank Dr. Gabriel just now, who introduced us on the methods on how to culture them. And also Dr. Claudia at the very beginning, who introduced us the application of this phytoplankton. So this phytoplankton, actually, you can culture them in a very large quantity. I believe just now there's a lot of pictures from Dr. Gabriel. Also, this phytoplankton can also be fed to other live feed, to rotifers, to copepods, to artemia, to all the zooplanktons. And after that, they can also be directly fed to the bivalve as well. So these are some of the common species that we see in Malaysia for the aquaculture industry, for the hatcheries that we have. We have Nanochloropsis, we have Isochrysis, Corella, Tetraselmins, Catasaurus, Thalassosira, and Duniella. These are some of the common species that are already found in the industry, found in the hatchery. So what is my research? My research is to find some of these species from our local area, from our local river, lakes, from our coastal waters, so that we can use them to culture in our local hatcheries. Now, for the common practice in aquaculture is we will produce green water. Aquaculturists, the fish farmers, they will prepare the pond, and they will, what they say in their term is they will make the water color. They will color the water green. So they are going to prepare the pond. They are going to fertilize the pond with fertilizer, and they are going to put in the water. Then they are going to let it be under the sunlight and wait for the color to turn green. And that green color actually is the phytoplankton. And as you can see from the picture at the right-hand side, each pond will have a different color. Although it is the same fertilizer, it is the same water, it's the same sun, but then the color will be slightly different because a different species will dominate in that pond. And if that pond comes with a better species, then it will be a better growth for your fish larvae. So this phytoplankton actually not only is a first food for the fish larvae, it is also for promoting the growth of zooplankton, which is also a bigger live feed for the fish larvae to eat. So we are preparing snacks in the pond for the fish. But there's something that is risky about this, which is we cannot control which species is going to dominate during the blooming, the fertilizing. So there's a risk of harmful algal bloom also. What if you don't have the right species in your pond? Then it will be harmful for your species. So that starts my first research grant in this institution, to look at the live feed, the potential live feed that we have in our local Sarawak. So this is the state that I'm in. So this is Malaysia on the right-hand side. This is West Malaysia. We have a part on the west side and an east part here. So I'm from this state, Sarawak, and my location is here in the yellow color. So we went over Sarawak to various places, and we collect this hatchery data from the fisheries agencies. And we found out that actually we have quite a lot of hatcheries, and most of them are focused on the western side or the southern side of Sarawak, which is this area. And when we contact them, most of them, they do not give us any feedback. They do not answer our call. They do not reply our emails. So we are not sure whether they are still active or not, or whether they have just registered with the government and then they the company shut down. So we are not sure. But then after surveying for so long, we found out that there are four hatcheries that use live feed. So we are excited to know what kind of live feed do they use. So I will show you the live feed products in the next slide. So here we see that there are people who tell us that they are not using any live feed at all. They are not, they don't have the techniques to keep the culture, to get the cultures. And most of them will just use a product. So these are some of the products that the aquaculturists are using. They are buying all these products from other countries, and most of them are not phytoplankton related. Okay, they are coming from squid, they are coming from fish meal, and so on. So that gives me the passion to start to find this gap already, and we would like to start something here. So I would like to start working on live feed, producing live feed, and then try to give this live feed to our local hatcheries so that they can improve their production, they can improve their survival rate, and they can improve their quality. So if live feed is so important, right, why isn't it widely used in our state, for example? Because it is very hard. It needs a lot of technique in order for you to get the culture. And then we cannot get the consistent availability, the volume that we need, because it's very hard to keep them alive. And also when we see just now from Dr. Gabriel's slide, you see it is exposed to the environment. So there is this matter of contamination from organisms, from other organisms, and also disease. And when you introduce this to your hatchery, then the disease will be also introduced to the hatchery. Also, I saw a question about harvesting, right? Harvesting is also something very challenging for this industry because of this small size. And then larval preference, the acceptability of this microalgae is different from species to species. It's just like I might not like broccoli and you might not like spinach. And then also the digestibility of the larvae, because they are still small and their digestive system might not be able to digest everything that we give them. And finally, meeting the energy and nutritional requirements of each species is different, because you cannot use one phytoplankton species to feed the fish larvae, it is not healthy for them. It is not a balanced diet. So we would like to have I have this vision that we will prepare live feed, just like the baby formula that we have now. Step 1 for newborn, and then step 2 for 3 days to 10 days old, and then step 3 for 10 days old to 15 days, and so on, so that the fish larvae will get all the nutrition that they need to get the best growth. Now, our government, actually our fisheries department, they have all these publications on live feed. Okay, the first one is the Manual of Breeding and Rearing Red Tilapia in Cages. But in that book, they also mention about producing live feed, for, which is green water for the tilapia fries. The second book about hybrid grouper, breeding and culturing. When it involves breeding, then always there is this live food. So draw your attention to this one, live food for fish. So it also introduces you all sort of zooplankton and phytoplankton for live feed. So if you're interested in all these books, please contact me. I think it's about time for me to end. So let me show you around my institution. So this is UPM Bintulu, an aerial view of our campus. And I am right now here in my office. This is the whole campus, where we have all sorts of, from cow rearing, chicken rearing, goat, and so on. Actually, our university is focused on agriculture. So we have all sorts of plants, we have animals, and of course, aquaculture, fish. So this is my field. Now, in this campus, we have 2 faculties, and I'm in this faculty, the Agriculture and Forestry faculty. And this is my department, which my office is up there. This is also part of our campus, where we have a central academic complex, where we have a veterinary lab, genetic lab, and so on. And this is in my building now. We have a teaching lab for teaching students. There are practical sessions. And also on the left-hand side, the aquatic biology lab is my lab currently, which I try to culture my microalgae and some of my aquatic plants. So on the right-hand side is another aquatic biotech laboratory of my colleagues. And we have also microbe labs, vet lab, genetic labs, wet lab, and histopathology lab. And I'm also affiliated with International Institute of Aquaculture and Aquatic Sciences. This institute is situated at West Malaysia at another location. So I will also show you the facility that they have. They have a few hatcheries there. They have a research boat because it is situated right beside the sea. And there are also microalgae lab, which you can see on the top left. Yeah. Other research lab, and then seaweed culture at the bottom, and postgraduate room for students who are attached to this institute or this laboratory. There is also guest house if you are there to visit, and here you can see it is right beside the Malacca Strait. So the niche of this institute is all this. There's a lot of different types of research, and I'm more related to the microalgae part. So these are the microalgae bank that we have, the species that we have here. And I think that's all from me. So thank you very much. Thank you for listening. And please leave me a message or email if you are interested to collaborate. Thank you very much. Back to you, Professor Bulan.
Dr. Tan, thank you very much for your very nice presentation and information about your institute. I believe that INOC member states researchers will definitely contact you for in order to get more information about your research, and maybe you can create a collaborative joint project opportunities. Thank you once again for your presentation. And I would like to invite last presenter, who is Dr. Rahul Vijay Kapoor. Before his presentation, I would like to give you information about his academic background. Rahul's research focuses on developing an optimized workflow for algal biotechnology for high-value and natural products, circular economy approaches, metabolome profiling of cells, co-culture techniques, microbial consortia, microalgal biotechnology, infochemicals characterization, microalgal biostimulants, biosurfactants, nutrient remediation, and resource recovery. He is research scientist for the deu.PSA project funded by Saudi Ministry of Environment, Water and Agriculture through Coast Beacon Development. This is a strategic Saudi governmental project related to food security, which aligns with Vision 2030 and the Saudi Green Initiative. Rahul is the laboratory lead and is mainly responsible for the fractionation and characterization of algal biomass and later development of algal biomass into commercial products. Floor is yours, Rahul.
Thank you so much for giving me an opportunity to present on this important aspect. And I would like to thank INOC for organizing this wonderful symposium. Well, we have already covered a lot of things related to microalgae, and earlier presenters have briefly covered what are the applications of microalgae. So my first slide is why microalgal biotechnology matters, and it's wonderfully covered that they have several applications of algal biotechnology. The first one is food and feed, bioremediation, CO2 sequestration, biofuels and energy, and then we go for some high-value metabolites. Okay, so that can be some phytohormones that can be used as a biostimulants for agricultural purposes. And we can go for cosmeceuticals and pharmaceutical products. Now, I've recently seen on chat as well, we were talking about macrolides, which act as an antibiotic, or we can extract anti-cancer metabolites from the algae. So these are really high-value metabolites, which represents some other side, which is not largely explored yet. So why this microalgal biotechnology matters? Because it's mainly aligns with net zero strategy, which is build, spread, greener. And more importantly, it aligns with expertise which is related to United Nations Sustainable Development Goals. Number 2, number 3, 13, 14, and 15. Overall, green recovery towards net zero, that's tick. The main focus that for microalgal biotechnology is we are not focusing much on the applied research. It's mainly based on the lab-based research, and we need to focus more on the applied research. And in order for the development, we need to have a core facilities where we can characterize all the different variables and what are the influence of those on the biochemical and molecular composition of the produced biomass. So this is one of the snapshot of the labs we have at deu.PSA project in Saudi Arabia. As you can see, we have some plant lab where they produce the microalgae, as Gabriel, Claudio, Emna already covered this aspect. And we have a lab over there where we do the basic parameter monitoring. Then we have preparatory labs where we extract the metabolites. And then we have core labs where all the mass specs are situated, where we do the absolute quantification of the metabolites. Right, so I'm going to talk about few analytical skills here. What is the most important is we need to have absolute quantification, and the minimum amount of biomass required is always a good opportunity when you have a lot of experiment where there is a very small amount of biomass available, mostly lab-based research, okay? So first one, we have techniques where we can quantify macromolecules such as proteins, carbohydrates, and pigments. Then I'm going to cover a method for fatty acid quantification and different fatty acid classes. And then I'm going to talk about what is metabolome analysis. Okay, as you can see here, we can quantify like 11 different metabolite classes, and they can be used along with bioinformatic tools to give a snapshot of how different variables are affecting different biomolecules when you harvest the microalgal biomass. So for micromolecules, generally we use 1 milligram pellet or biomass or the freeze-dried pellet, and we use that one to quantify proteins and carbohydrates and pigments together. So the carbohydrates are quantified using an Anthron method. As you can see, these are the glucose standards, and we use a standard curve. And then the proteins are quantified using BCA method. Okay, as you can see, this is the concentration range. And then you can use this equation to quantify the, concentration of proteins and carbs in your unknown sample. What are the other analytical techniques that we have here? So we can quantify the same macromolecules. The earlier one was spectrophotometric method, where you can use FTIR as well. Okay, and then FTIR, using FTIR, you can quantify the macromolecules. So what you need is a dry algal powder. Okay, other things that we need to know, what is the ash content? Okay, and you can use stable Moffel furnace that we have here, and you can then quantify the ash content. But you can see we are using very tiny volumes here. Okay, we are not using big volumes. And then you need to know what is the inside concentration of carbon and nitrogen inside the cells. So you can use CHN analyzer, or it's also called elemental analyzer. And in order to know the heavy metals, because algae is sometimes used for bioremediation purposes. So in order to know the heavy metal composition, you can use XRF, or if the concentration of heavy metals is really tiny, then you can use ICP-MS. Right, so next, going on a little bit from macromolecule to little bit molecular level. So why metabolomics actually important and how it gives feedback to the biosciences. So for example, if you can see, in omics, there are metabolomics, proteomics, transcriptomics, and genomics. So genomics exactly tells you what is possible. So when you have different conditions, the reaction, for example, microalgae, for example, if you increase the concentration of nutrients, or if you increase the light, okay, so it's a kind of a stressor, okay, and then microalgae reacts at a genomic level very late. It takes number of, iterations in order to react. In case of transcriptomics, it sounds like what appears to be happening. Whereas in case of proteomics, it sounds like what can make it happen. Whereas metabolomics, it reacts instantly. Okay, so that's why metabolomics directly gives feedback to the biosciences. So in short, metabolites, if you identify the metabolites, they are nothing but the canaries of the genome, and they represent the final omic level in a biological system. So you can say they are very quick to react to the stressors. So the stressors are different stressors such as biotic and abiotic. So these are biotic and these are abiotic stressors, and any response to that, if you want to capture, okay, you can use metabolomic study, or you can use all the biochemical and macromolecular composition, and you will be able to assess which variables are better in order to improve the composition of specific compounds in your microalgal or algal biomass. So this is like a general protocol for metabolomics. I'm not going to go in detail because in order to cover the analytical platform, you need a separate presentations, but I'm going to give you a snapshot of what metabolomics is. In metabolomics, first thing is as soon as you remove the culture, you need to quench the culture. Now, quench means metabolome is so dynamic. For example, ATP, its turnover time of changing is less than a second or millisecond. So what happens, you need to, as soon as you take the culture out, you need to quench the biomass in order to stop the metabolic activities. And you can do that via quenching. Okay, quenching means you can use, say, 60% aqueous methanol, okay, but at a very low temperature. I'll say minus 80 degree or something. And you need to quench the biomass, and then you need to extract that biomass. Then the extract, then you can use the either GCMS or LCMS. And there are a lot of different data analysis platforms where you need to combine the bioinformatic tools as well. You might need to use MATLAB or R platform. You cannot just simply use Excel to do the MS data analysis. And then you need to map those upregulated or downregulated metabolites using data analysis platforms. Okay, so you can use KEGG map pathway, or there are a lot of other tools now available where you can map all these metabolites and can understand what is happening inside the cells. Next, fatty acid analysis. As you can see, it's very important. A lot of speakers already covered, like, importance of fatty acids from microalgal biomass. And as you can see here, this is like a 37 fatty acid standard that you need to inject first. And you need to make sure you resolve all the peaks correctly. And then what you can do in order to quantify, you need to have a right strategy to make sure all your quantification of all the fatty acids is absolutely correct. Okay, it's very important any variables that you're using in your culturing. For example, you want to change nitrogen, you want to change phosphorus, you want to change the CO2 concentration, you want to change the pH or temperature or the light irradiance. So they have direct influence on the concentration of these molecules. So what we do is we generally use like a different internal standard. You need to make sure the internal standard or fatty acid that you're using is not present in your biomass. If it's present, it's gonna give you completely wrong quantification, and that can be misleading on the large scale or the lab scale research. So in generally we use C13, C17, but it can be variable depending upon the research and depending upon the microalgal species that is under investigation. So how you can report this fatty acid data? You can actually classify them as like, first thing is like you can report them as a total fatty acid. This can be a conditions. And then you can divide them into saturated, monounsaturated, and polyunsaturated. In our case, I think the high value products for us are the EP and DHA, which is total unsaturated fatty acids. There is another way to report is, for example, imagine there are two conditions and the total fatty acid concentration is 6.83% dry weight. Then what you can do, you can actually work it out. Individual fatty acids, what is the percentage of total film? So for example, the C18, which is linoleic acid, is actually 35% of the total fatty acid. So there are different ways of reporting. What is most important is the method that you're using is quantifying it accurately. The method of quantification should be reliable, and there should be a good amount of replication. Minimum, it should be 3. Now, these are all the techniques. I'm not going to go in detail of individual technique. There are a lot of aspects, but I want to give you an example. When you have proteins, carbohydrates, you have internal carbon and nitrogen levels, then you have detected fatty acids, all the metabolites, how you can use this data actually for the research, right? So let me give you an example of blue sky research. So what is blue sky research? Is something you have a hypothesis and you want to test it. Whereas completely opposite research example is the applied research, where we actually go for the applied research with a particular target here. Okay, so this is a blue sky research example. And now imagine you have different variables such as carbon, nitrogen, phosphorus, and you want to determine what is the influence of these components on accumulation of, on this side, you can see total fatty acids, TFA. What is the influence on the carbs? What is the influence on the proteins? And what is the influence on the EPA, which is eicosapentaenoic acid, which is nothing but a high value fatty acid, right? And these are the counterplots. So let's look at, if you're interested in TFA, you can apply this to any of your research. So if you're interested in total fatty acid, for example, which is A to C, right? So you can see carbon and nitrogen here, the plot A, the counterplot. You can see is the ratio of C to P and N to P. So in order to have a higher total fatty acid content, the red represents the higher concentration. So C has to be high. As you can see here, this is carbon and it's 2 to 60. So in order to achieve the higher total fatty acid concentration, your C needs to be higher. As you can see, it's represented by the red color. Whereas what is the situation with the N? The N needs to be what? Low. Okay. Then let's look at the P. What it says, the P is not a major factor because it's not giving exact correlation. So in general, if you want to achieve higher fatty acid, your C should be high, N should be low. Let's look at some other important component. If you are interested in carbohydrates, for example, look at counterplots D, E, and F, then P has to be low. So let's look at here, which is P here, okay? And then you need to have, if the P is depleted, then it leads to the accumulation of the carbohydrates, okay? And then you need to have, when N gets depleted also, there is an increase in the C to N concentration. You can apply this for proteins, you can apply this for EPA. For example, let's look at the proteins. For example, in order to have a higher protein content, you need to have high N, which is here, as you can see, okay, and you need to have high P, okay? So it varies to a different degree, but this kind of analysis gives a clear snapshot that what are the variables. You need to remember, we are just analyzing C, N, and P variables here for accumulation of different compounds. In doing so, you need to make sure the other parameters such as temperature, pH, light, and other things are kept absolutely constant. Now, this kind of analysis can be used for applied research as well, provided you are harvesting the microalgal biomass at a larger scale using the same seasons. So you need to have some seasonal data. So the temperature and the light irradiance can be somewhat similar. So if you harvest in March, then you need to have like March to April or April to May, okay? You can correlate like that. Let's look at another example, how it can be use all these macromolecular composition and molecular composition for applied research. So we worked on this one of the project called ALG-AD in UK. And then we have used the anaerobic digestion waste, okay? It's called digestate, to grow the algae. And then that algae was used for animal feed. Okay, so it's kind of a good example of a circular economy. But then we use this biomass for the fish feed. And now here you can see, we can go deep down into a little bit of detailed analysis on omega-3 and omega-6 fatty acid. And what is important, the ratio of omega-3 to omega-6 should be 1. If the ratio is 1, then it lowers the risk of developing any allergies or inflammatory condition or cardiovascular disease. So what I mean by that is when you have biomass which is grown either using a circular economy approach or using a lab-based research or using a large-scale cultivation, and if you want to use this biomass as a fish feed, okay, then when you analyze this fish feed compared to your control conditions where you're feeding your normal diet, you need to make sure the fatty acid composition in the flesh and the ratio of omega-3 to omega-6, if it's coming around 1, then it's really good results in terms of either bioavailability of fatty acids that you're providing through your algae, and then it's getting absorbed in the fish, and in the fish flesh composition is represented here. So as you can see, this is a commercial diet, and the ratio of 3 to 6 is above 1. Okay, whereas when commercial and algal diet is combined, the ratio is brought back down from around 1.5 to around 1, as you can see here. So this is the importance that we need to keep these analytical platforms in completely reliable way to quantify at different stages of microalgal biotechnology. Let's look at some another example very quickly. So this was about the fish feed. Now let's talk about the animal feed. Either you can feed it for the cow. For example, as we know, there are different ways of downstream processing, as Emna has already covered. Yet you can feed directly the biomass without any processing, or you can use hydrolysis, or there are a number of processes that you can apply in downstream processes. For example, in this case, if you hydrolyze the biomass, then what you get is a free amino acid and some bioactive peptides. So the peptides which have low molecular weight, okay, are known to have good bioavailability. What it means, imagine if I'm eating a fish, for example, and there are two different varieties of fish. One fish contains some lower peptides, which has good bioavailability. It means it will get absorbed by the body, okay? And another fish, for example, the protein concentration is good, but the peptides that are available, they are not broken down properly by the digestive system. So they are not bioavailable. So when we did the hydrolysis of the microalgal biomass, as you can see, molecular weights, the highest concentration of the peptides that we have in Cynodesmus, for example, they are low molecular weight. So then it also confirms like, yes, all the large scale cultivation, all the hydrolysis process, and then the fish feed trials and the animal feed trials are working, and this gives confirmation. So this is certificate of analysis is really important in this case, okay? And if you have the low molecular weight peptide, they are more bioactive and it improves the health and immune system. Right. Another example of blue sky research, this is the last example, is like how you can use for these analytical skills for microalgal consortia. So for example, if algae and yeast, they might be beneficial to each other, or they might not be beneficial to each other. How? For different biomanufacturing routes. So for example, algae can give something to yeast, yeast can give something to algae. Same like there is algae and bacteria consortium. Okay, so we want to know what is the interaction between these communities, and it's very essential to trap them correctly using analytical techniques. So for example, just look at here, for example, this is yeast and algae example. This is on 0 day and this is on 9th day. And as you can see, algae is at the center, whereas yeast is inoculated on the sides. So what we do is we harvest here into the space. These are the agar plugs, we extract them. Then if you want to know the interaction between yeast and yeast, you can harvest here. If you want to know the interaction between yeast and algae, you can harvest here. Okay, and these are green are the upregulated metabolites, whereas the red ones are the downregulated metabolites. You can use this data, map it on pathway, and you will be able to quickly understand if the consortium is actually helping to achieve the desired product, or it's actually antagonist, or it's not actually beneficial to achieve the desired product. Okay, so these are the different ways for blue sky and applied research. You can use the molecular techniques, okay, in order to understand what is the interaction. These are there, you can check on my papers, but these are the papers related to microalgal consortia or microbial consortia. And thank you so much for listening, and thank you so much for giving opportunity to talk on this important aspect. Thank you.
Dr. Rahul, thank you very much for your very nice presentation and information. You have one question from Levent. Levent, would you like to ask your question?
Thank you very much. Dear Dr. Kapore, thank you very much for your nice presentation. It was amazing. I like it so much. Related to the bioactive peptides, we are currently studying on the bioactive peptides from the algae. So I'm asking you, if you apply proteases in your lab on the biomass to obtain the bioactive peptides from the microalgae or macroalgae.
We have used there, as you know, like there are different types of proteases available. So we have tested a lot of different types of proteases for different strains, because it mainly governs by which microalgal species you're working on, and what are the mainly peptides that you're interested. So I think in UK we have used the neutral proteases as well, and that was used for hydrolysis and to get the bioactive peptides.
Okay, thank you very much.
It seems that we don't have any other question from the participants. I think we came to end of the 7th version of latest developments on marine sciences symposium. And before closing, I would like to highlight that INOC will be more than happy if you can collaborate and cooperate in research areas of common interest with a view to help each other in building capability in the field, and of course exchange information and maintain dialogue in the developments in the field of mutual benefits and use. And please propose your joint projects of research and development of common interest to member countries of the INOC network. And please help and assist in training of quality manpower for the INOC member countries in order to development. So before closing, I would like to thank two important persons. One of them is Ong Meng Chuan from University of Malaysia, and another one is Mümtaz Trashan from King Abdullah University of Science and Technology, KAUST. These two people made important contribution for organization of this symposium, INOC symposium. So thank you, Ong Meng Chuan, and thank you, Mümtaz Trashan. So the next thanks goes to our keynote speakers. I would like to show their certificates. Let me share my screen in order to show your certificate. Dr. Claudia, Dr. Ergün, Dr. Tang, Dr. Emna, Dr. Levent, Dr. Gabriel, Dr. Tan, and Dr. Rahul. Thank you very much for your very nice presentation and information for Inter-Islamic Network on Oceanography member states researchers. I believe that this your presentation, the information you provided in your presentation will bring more opportunities in near future. So before closing, I would like to also thank INOC President, Professor Dr. Şermin Açık Çınar. Without without her help and support and contribution, it is almost impossible for organization of this kind of symposium. So we really came to end of this symposium. Thank you very much once again for your presentation and information. And I am looking forward to seeing you in other INOC events in near future. In order to get more information about INOC facilities, I mean symposium and educational events, please don't forget to visit our INOC website and INOC social media accounts. So thank you very much once again, and hope to see you in near INOC facilities. Bye-bye from Turkey.
Thank you very much.
Thank
Bye-bye.
you. Bye.
Bye.
Thank you.